Hello and welcome to another RLabs tutorial where we will discuss the function called aggregate. Aggregate is actually very similar to the tapply function, which we talked about in the previous tutorial, but it can actually accomplish more than tapply. Now there are multiple ways to use the aggregate function, but I'm just going to show you one straightforward way, my favorite way, whose general form is shown here. Now this method only works if your data is in the form of a data frame or a list. So if your data is stored in independent vectors, you'll have to coerce them into a list or a data frame first, which is simple enough anyways. So the function takes a minimum of 3 arguments. First we have a formula which takes the form of y tilde x, where y is a variable, typically a numeric variable, that we want to divide up into groups according to x, the grouping variable. Next, we specify the name of the data frame that contains x and y, or the list that contains x and y. And then we specify the function that we want to apply to each of the subsets of x. And there are function-specific arguments that we can indicate at the end of the function. So to demonstrate how aggregate works, we will use a data set that is built into R called CO2. And these data were obtained from an experiment investigating the cold tolerance of this grass species here, whose name I'm not going to try to pronounce, but there it is pictured here. 12 individual plants are included in this small data set, 6 from Quebec and 6 from Mississippi. And at each location, 3 of the 6 plants were assigned to a control group, the non-chilled group, and the other 3 were subjected to daytime cooling. Then at 7 different ambient CO2 levels, the CO2 uptake of each plant was determined and recorded. So the initial data set contains 5 columns, one for the individual plant IDs, one indicating the type of plant, i.e. the location of origin, one indicating the treatment to which the individual was subjected, another one indicating the ambient CO2 concentration level in milliliter per liter, and finally a column containing the CO2 uptake data in micromoles per meter squared second. Now before filming this video, I actually made up and added another column for demonstration purposes called height, and this column represents the reproductive plant height in centimeters. So you can see that the dataset is already in my global environment because I have already called up the built-in dataset and added in a column. So here it is, and you can see that we have 84 rows of entries and 6 columns. Now let's say that we are interested in looking only at concentration and uptake for a second. So we want to ignore all other columns and just find the mean uptake at an ambient concentration of 95 milliliter per liter, the mean uptake at an ambient concentration of 175 milliliters per liter, and then at concentrations of 250, 350, 500, 675, and 1000 milliliter per liter. Well, the aggregate function can help us here. Before the tilde, we specify the uptake column because this column contains the values to which we want to perform a function. Then after the tilde, we specify the concentration column since this column contains the 7 categories that we will use to subset the uptake values. And we also have to indicate that these columns are from the CO2 data frame. And then finally, we specify that we want to take the mean of each of the subsets of the uptake values. And there we go. Now, if you recall from the previous video, we can also use the tapply function to do the same thing. You can see that we get the same answer. We get the mean associated with each of the 7 concentrations, the only difference being the format of the output. But the benefit of aggregate is that it can go above and beyond what tapply can do. So let's say, for example, that we still want to subset by concentration, but we also want to further subset by treatment type. So recall that we have 7 different concentration levels and 2 different treatments, and we want to know the average uptake by non-chilled plants at an ambient concentration of 95 milliliters per liter, and of the chilled plants at an ambient concentration of 95 milliliters per liter, and then of the non-chilled plants at an ambient concentration of 175 milliliters per liter, and of the chilled plants at an ambient concentration of 175 milliliter per liter, and so on and so forth. Well, tapply can't handle multiple subsets, but aggregate can. So remember, here's the code that we use to subset the data by concentration alone. And if we want to subset by treatment as well, all we have to do is literally add it on to the list. And we can subset by more than just 2 columns as well. Here we have added type to the list of columns by which we want to subset. Now, if we want to subset in the same way but get the means of more than just the uptake values, for example, if we want to get the mean plant heights as well, we cannot simply make an additive list like we did with the subset columns. What we have to do is C-bind or column bind the appropriate columns, i.e. the columns containing the values to which functions will be performed. So in our case, we have to C-bind uptake and height. So here we have subset the data by just concentration alone, and we have the uptake and the height of each subset group. Now, just to show you that aggregate still works on list, here's the same data in list form. So we have all of the plant IDs and types and treatments and concentrations and uptake values and height measurements. And now we can execute that last aggregate function that we just saw, but now instead of having CO2 as the specified data frame, we have CO2.list as the specified list. And there we have it. Now I know that I have used mean as the function in all of these examples, but just to clarify, it does not need to be the mean function. We could, for example, use length to determine how many entries are in each subset. So we'll use this simple example here. We want to find the length of subsets within the list called CO2.list. We are using the groups in the concentration vector to subset the data, and we want to find the length of the uptake value. So we want to know how many uptake measurements were taken at a concentration of 95 milliliters per liter, at a concentration of 175, and so on and so forth. And you can see that there are 12 values in each group. Now note that instead of uptake, we could have specified any other vector from the list in its place if we wanted to, such as height, or type, etc. And we get the same answers because all of these vectors contained within CO2.list have the same number of elements, and therefore would all be subsetted in the same way. Thanks for listening to this RLabs tutorial video. If you found this useful, you might be interested in our free online Moodle course, or check out and subscribe to our YouTube channel.